My name is Jaden Bowerman, and it is 5/11/22. And I will start with case 1 today. The blood panel shows elevated and actively elevating blood ammonia. Orotic acid levels were also elevated, and her citrulline levels were within normal limits. We'll begin with outlining the normal inner workings of a nitrogen within the body. Overall, the movement of nitrogen is in the order of transamination, oxidative deamination, and either excretion through the urea cycle or use as a substrate for the synthesis of other nitrogen-containing molecules. Transamination is the transfer of an alpha amino group from the amino acid to a nitrogen-accepting group, such as alpha-ketoglutarate. This transfer reaction produces an alpha-keto acid, which is useful in synthesizing high-energy molecules, such as pyruvate, for use in gluconeogenesis. The reaction also produces a more usable amino acid, such as glutamate, that carries ammonia throughout the body. Ammonia is a toxic molecule by itself to the body, so we have non-toxic carriers to transport it to different tissues as needed, hence glutamate. It was the product of the transamination process before, and it is a fine nitrogen carrier, but it can be further modified into a better nitrogen carrier called glutamine, which holds 2 nitrogen groups. Glutamine is still non-toxic, and the enzyme responsible for this reaction is called glutamine synthetase. While glutamine is a great carrier of nitrogen, it cannot hold on to it forever, and that is where glutaminase comes into play and removes one nitrogen group for the use in the urea cycle. The remaining product is glutamate, which has 1 nitrogen group remaining to be removed. And oxidative deamination will free that last nitrogen group as ammonia by use of glutamate dehydrogenase, as well as reducing equivalents like NAD+ and NADP+. This is a reversible reaction that is dependent on cellular needs, the ratios of those reducing equivalents, and the pre-existing amount of free ammonia in the body. So I briefly touched on it before, but nitrogen transport is useful in more than just removing ammonia. Nitrogen transport system during the transamination process produces carbon skeletons. When alanine undergoes transamination with alpha-ketoglutarate, the products are pyruvate and glutamate. Pyruvate is the carbon skeleton, and it's very useful in the fasted state as a substrate for the gluconeogenesis pathway in specific. So now that we understand what is supposed to happen, we can talk about our patient with the crazy high blood ammonia and orotic acid levels. If ammonia levels are high, we know it is being made and transported properly. Her high blood ammonia levels are indicative of a faulty urea cycle, because that is where ammonia is transported to and prepared for excretion. We can then pinpoint where the urea cycle is deficient with the increased orotic acid levels and normal citrulline levels. Ammonia enters the mitochondrial side of the urea cycle. This portion is highly regulated by carbamyl phosphate synthetase 1, which produces carbamyl phosphate. That product would then react with ornithine to produce citrulline by an enzyme called ornithine carbamyl transferase. If this enzyme is deficient, which I suspect it is, there would be an overproduction of carbamyl phosphate. This is used in pyrimidine synthesis, but the intermediate step for the synthesis is the synthesis of orotic acid. Her citrulline value is just below the normal values, which in this case helps justify the idea that ornithine carbamyl transferase enzyme is deficient. Also seen with this deficiency is decreased aspartate synthesis within this cycle. The decreasing overall quantities of the non-essential amino acid would result in its character becoming closer to that of an essential amino acid, The enzyme that is not in question is arginosuccinate synthetase. If this enzyme were deficient, we would see an overproduction of citrulline, as that is the step after citrulline is produced, and it would then be impaired. So now we have case 4. We have a patient with exercise intolerance, tingling in the extremities. She's tachycardic, a heart murmur, and a suspected megaloblastic anemia all are working against her. So I wanted to talk about vitamin B12 first, as this is what I came across as the cause when I was researching the condition. Vitamin B12 is used in purine production as a carbon and nitrogen donator for the synthesis of the second ring. It turns out that purine is put to use to start cell differentiation after erythropoiesis takes place. Without sufficient purine molecules to elicit this differentiation process, the red blood cells become abnormal in size and shape, as seen with this condition. Iron was the next option, and it is needed for red blood cells, mostly for hemoglobin production. It is, though, possible that her iron is low due to low hematocrit values and her progressive fatigue. When red blood cells are broken down at the end of their days, the iron is recycled for reuse. So an iron deficiency alone does not account for the rest of our patients presenting symptoms. It would not be a likely choice. Vitamin B6 aids in the production of hemoglobin as well. It is also used in amino acid metabolism and gluconeogenesis, and it transports other molecules around the body as a water-soluble vitamin. If deficient, these synthetic and transport pathways would be worse for wear. The cysteine pathway is one in specific that would be impaired from turning homocysteine into the intermediates for the TCA cycle, gluconeogenesis, and liver excretory pathways. A complete blood count and blood smear or stain would be incredibly beneficial to confirm or deny the suspected deficiencies. These two tests would confirm the number and morphology of the blood cells present. And as I was doing some research, a methylmalonylic acid test is used to test for vitamin B12 deficiency specifically, as it's a cofactor for the synthesis of succinyl-CoA from methylmalonylic acid. If it's deficient, the methylmalonylic acid value would be elevated without the possibility of being turned into succinyl-CoA. And that is all I have for this one. Thanks so much.